Such a popular seat. Everyone's trying to sit
The
in my
only one
seat.
left.
I picked the best seat in the house.
Bobby, do you want me to move my stuff?
Who? I know. I need like access, but I don't want to be in like the front row because I don't see very well. I'm old. Okay, so I'll get started. Thanks everyone for coming. It's my pleasure today to introduce Dr. Isaac Esquivel. Isaac came from California, then went to Texas, and now is in Florida. So he likes to be in warm places. I know Isaac because we were in the same cohort in our PhD programs. We met in something that was a very traumatic experience for me and became friends during a statistics class that you signed up for the earlier session, so I would try and get answers from him from the exam because he took it first. So he's kind of moved about the southern states, and now he's an extension specialist and assistant professor in the University of Florida. I think he's going to introduce himself, so that's all I'll say. But thank you for coming.
All right, thanks, Chloe. Thanks, Chloe, for the introduction and invitation to come speak to you.
One more thing. Isaac's like right on time with his talk, so I'm going to ask that there be no questions until the end. Otherwise, he'll be here for 2 hours.
Yeah, I have a notorious for going over time, but this should be fine. But yeah, so generally, I'm assistant professor, extension specialist, University of Florida, the NFREC. And you may be wondering why the title is talking about bees when I'm sort of the agronomic forage crop extension specialist for the Panhandle. And that's basically I cover mainly focusing cotton, peanut, pretty much all the row crops, corn, soybean, and then forages, so pastures, small grains, oats, things like that. But I'm going to sort of give you a little background on how I got to bees in the system. It's going to be throughout the talk, but I did undergrad at UC Riverside, where I really loved like Hymenoptera. So I was databasing parasitoid insects in a lab for a while. Then I moved to sort of research for undergraduate research, looking at tomato psyllid and potato psyllid in peppers and tomato. Then grad school-wise is where I went to A&M, and I was mainly tasked at looking at the spatial relationships of cotton pests in South Texas cotton systems. So without telling too much backstory, let's get this going because it is just about 45 minutes. So here's just a general brief outline of how we're going to go. So just broad research interests and background, give you a little background on that. Talk about pests in the cotton agro ecosystem because that's sort of where I initially started before I got into bees. Then do a little brief crop pollination intro for those not familiar with pollination systems or pollination in general. Then talk about pollinator independent cropping agro ecosystems. Then we get into a little bit of bees and cotton and other pollinator independent crops. Then the benefits to pollinator independent crops from bees and potential benefits from the crops to the bees. So that's where that whole quid pro quo sort of verbiage comes from. So let's just get into my research interests. I have generally like relatively broad research interests, and I think I actually saw this slide from Cesar, who gave a talk at an extension meeting at one of Chloe's symposiums a few years ago, and I sort of liked the layout and sort of adopted it and changed things for my research interests. So generally, I'm broadly interested in studying the role of biodiversity, whether that's pest herbivores, pollinators, beneficial insects, partially governed by landscape structure, so how the surrounding landscapes interacts with this. But in ultimately, so I'm using this, using sort of the studies we do in this system to develop improved strategies for pest management strategies for agricultural systems. So if you're just looking at this layout, so we have whole agro ecosystem over here on the left-hand side. We'll sort of define it in the next slide, but we have different layers of this agro ecosystem. So one starting at the very bottom, at the crop level. So those are things are getting to not even the insect itself, but just in the crop. So whether you're having soil type differences on insects or pretty much anything, fertility issues, other abiotic factors, variety issues, or different varieties, cultivars, new crop traits. For example, I work in cotton. We have a new BT cotton variety that's commercially available this year, and it provides protection against thrips and stink bugs, or plant bugs, not stink bugs. So it's going to be interesting when we start planting a lot more of that. That's sort of going to change how the relationship of thrips in the system, or especially in the Panhandle, because we have cotton and peanut, in which the thrips are pests on both. So if they're not going to be attracted to cotton because of that variety, what is that going to look like for peanuts in the system? Then we get to the middle level, sort of the field level. That's where we start getting into the pest management. Within field pest management, I'm really interested in sort of the spatial variation within a field and how we can improve insecticide application strategies on the field level, but also looking for improved thresholds, pest monitoring, scouting efforts for our growers in the region. Then when we get to the top, getting to landscape level approaches, where we start getting into just through things like diversity, whether it's species diversity, landscape level diversity, and then what that means for sort of regional pest management across, not only regionally, like say the Florida Panhandle, but maybe in the Southeast more in general. So you can see all these relationships sort of have, there's going to be effects from or to these things, from pests, natural enemies, pollinators, which ultimately have effects on yield, fruit quality, ecosystem services, like say pollination, natural biological control from natural enemies and predators. So what is an agroecosystem? Sort of like a fancy term. You hear it a lot. I use it a lot. It's a lot in a lot of the literature.
Your screen's not being shared.
Oh, it's not being shared. How do we do that?
Sorry.
Yeah, go.
Pause. Did you just skip a slide?
No,
Because I thought I saw like a flash. Okay,
I think it's just the
that's all.
projector.
Thanks. Make it big. No? Okay. How do zoom open?
I think it's the corner.
The corner.
The corner. The
Oh
left-
no, go back to the window with Lisa Reed.
Yeah.
See
that-
the bottom right?
Ah, thank you. So glad we have young students to tell
That's
us
the Mac
how to
system too, so I
do.
haven't used Mac in a long time.
The most of your.
We should get the little, okay.
Okay, yeah, that's
And the comments are all, can't see the slides. Okay.
should be good now.
Okay. All right.
Okay,
Sorry.
and we're back. No problem, I'm not muted. Okay, we're good. Okay, so what is an agroecosystem? I use it a lot. It's in a lot of the literature, especially when we're talking about these systems or things at different levels. So just basic definition, it's a functioning, spatially explicit area of agricultural activity within a landscape. So it includes their living, non-living components and their interactions. So basically it's agriculture plus ecosystems. So study of ecology in an agricultural setting. And I really like this diagram. I think it gives it a good show. I added some pictures of it so we actually have some context. But it's very sort of people-centric, so say people at the center. Our management practices, whether it's on individual taxa, like pest species, or sort of, say, using tillage, in which we change the landscape. So there's a lot of landscape fragmentation. For example, this is a study system we'll be talking about, the picture there, of a cotton system in South Texas. And then all that's going to affect sort of our ecological processes, whether it's pollination or natural biological control, which has effects on, they use ecosystem services, or they use ecological services. I think we use ecosystem services, sort of the better term, and that ultimately comes back to us and has effects us on us. So I'm sort of interested in studying all these relationships, how they work together, and how they ultimately affect us. Now, yeah, use the mouse. Okay, now keep going. So when I first started, I was really, one of my co-advisors was Bob Coulson. He was really big on sort of landscape ecology, pine beetles back in the day. So we're looking at using landscape ecology in sort of my program in cotton systems. So I think I started getting more, well, how does the landscape affect things at the local scale? So landscape structure and composition, do they affect local scale processes like pest densities and eventually pollination, but also biological control from natural enemies already present in the system. So this is just an example of the cotton system I work in. It's a relatively large cotton system. This picture on the left is one commercial operation down in South Texas. And that's just to give a sense of size, but I'll talk about it in a couple of slides as well. But that's just about 40,000 acres of cotton and sorghum in just that picture on the left-hand side alone. So these are relatively large fields, which is why I was interested in seeing how the pest dynamics within those fields were affected by landscape structure. So getting started, what was I looking at in these cotton systems? So the main pests of focus in South Texas were cotton fleahopper, Verde plant bug. Now since boll weevil eradication and introduction of transgenic cotton varieties, or BT cotton, for bollworm and boll weevil control, we started using a lot less insecticides since those were established. And since we started using less insecticides, these sort of secondary pests that were at once a time taken care of by those sort of calendar weekly sprays or every other day sprays for boll weevil and bollworm became the forefront or the major the primary pests in the cotton system across the southeast. So South Texas, we had these two plant bugs. Is cotton fleahopper. It's sort of more of a Midwestern Texas, Mid-South species. And then verde plant bug is sort of endemic to the Texas and Mexican Gulf coasts, whereas in the southeast we have a lot of stink bugs and then tarnished plant bug. And they're both pests sort of on pre-bloom cotton, so they attack the fruiting structures before they flower and produce a boll, and their damage causes them to abscise, so you're going to be using yield in those early season, because generally the way cotton works, those first position squares or the first squares on the plant are the ones that produce most yield, so they're very important in sort of yield production or in boll production. Now, so I was interested in how does the landscape influence these pest populations, and sort of answer that we had to develop sort of sampling strategies, or main things where I was interested in are there edge effects, so are populations different at the edge of a field compared to field interior. Now when I mentioned that, say for example this field here on the left was about 1,200 acres, that's where that came into play. Is like, well, maybe not everything's the same across this sort of 1,000-acre field in terms of population density. And then I was interested in interface effects. Used to call them ecotones. That's more of a landscape ecology term, but we weren't really looking at true ecotones, which is a transition area between two different crop types. So I think adjacent or interfaces is the more appropriate term. So we're sort of seeing how does the adjacent crop or habitat influence pest populations in that area of the field or in a particular field. And then this is a digitized version of the image I was showing you all earlier of just our cotton system. And it was primarily cotton, sorghum on a rotation. So whatever you see that's red was cotton one year, it'll be sorghum the next year, and that just keeps going unless there's something going on in economics of crops or other issues. They'll swap to other crops. So does the landscape influence pest populations? So again, edge effects, interface effects, and I did two different sampling methods, one being just standardized transects, and we ran them across 5 fields. So we did 2 parallel transects at the edge of the field to the field interior up to 300 meters into that field. We sampled fields across 2 years, and then to sort of account for the spatial variability, as those are sort of structured different distances from the field edge, we also did random point sampling, in which we broke down that big field into little sections, and then we'd use a random point generator to generate points no closer than 75 meters apart, and that we got that number based off literature from Ligas, and that was their maximum dispersal rate in a given day, that they could travel 75 meters. So we wanted to make sure our points were within that 75 meter range to sort of account for that at least populations we found were independent from one another. So just getting straight into data, so we can talk about bees. So this is just an aggregation of all of our data for cotton fleahopper. The trends were similar for both species, but this is just all data across 2 years for cotton fleahopper. And we can see that pest density, there's a significant edge effect. Pest density decreases as you move from the edge of the field into the interior of the field, and sort of approaching 0 at that 200 meter mark. And here's just sort of or on the image on the left is this map to sort of show that. So we have reds, higher population density, closer to that semi-natural habitat, which is green, compared to we're getting towards 0 or maybe like 10% or 1 per plant in the field interior up to about 200 meters. Now, if you would take into account what's next to that cotton field, how does that matter? So not only does the pest density decrease from the edge of the field towards field's interior, but it's also influenced on what is next to it. And that is mainly driven by the interface of cotton semi-natural habitat, which is CN, so that the blue dots or the blue triangles in that blue line. So not only do we see more pest populations or higher pest populations in that area of the field, the rate of decline is also more strong in that area of the field as well. So if you look at the bars, you can see that overall rate of decrease is actually steeper and sort of approaches 0 between 150 and 200 meters into the field compared to the other interfaces of whether it was next to a cotton field or whether it was next to a sorghum field, where we have sort of a moderate population of pests, and they slowly decrease to the field, and that it extends towards 200 meters into the field. So it seems like they're going a little farther into the field in those areas compared to that semi-natural habitat border. Now, what does this mean sort of in terms of pest management? What's sort of the ultimate goal of sort of that project? So if we go ahead and lay the economic threshold for cotton fleahoppers and verded plant bug, which is about 0.1415 bugs per plant, you can see we're only really above threshold in this upper left quadrant, so that's areas of the field that are next to semi-natural habitat and areas of the field that are less than 100 meters from the edge of the field. So we just started thinking, it's like, well, is it really necessary to spray an entire 1,000-acre field when our pests seem to be in this one area of the field? So the ultimate goal was sort of to get our growers to think about different application methods, or we call it zone management. I think we use sort of precision ag now, sort of more of a precision insecticide application. And we never told them exactly to do this, because growers do not like you telling them you don't need to spray. They will get mad if you tell them, well, you don't need to spray that. So, and in the cotton system, insecticides are relatively cheap, especially for plant bugs. So they spray generally 1 to 5 times, depending on what pest it is and the availability of insecticide. So they're generally going to spray their field at least once. What we wanted to do was sort of convince them that maybe you didn't need to make that secondary application a week later, or if they were going out to put herbicide, they usually put an insecticide in the tank as well. So we never told them this, but we would give them sort of maps. We would show them this data, so we had a good working relationship with the management of that large commercial field, and we would give them our data every time we went out and sampled. And I'm going to show you images on that in a second, but I'm going to finish this little story. So after 2 years, and I was there for 5-6 years, can't remember now, but we were doing this not only in cotton, we were doing this in sorghum with aphids, worms, stink bugs, and sorghum as well. And we would show them the data, and then write a little report. And after 2 years, I was talking to one of the manager, and he was like, well, we were seeing your data, and we ended up just spraying half of the field, or the side of this field, for example, where there's more curvature. They went along that edge of the field, just stuck one side of the boom out and sprayed that field once. And it doesn't seem like a lot, but these sprayers they're using cover 20-30 rows at a time, so it's a pretty significant area. So they only sprayed that side of the field twice. So they did end up doing something. So I didn't appreciate that until my current position, because when you're in grad school, no one really teaches you about extension. So the main sort of goal or philosophy of extension is sort of to provide a change in practice, or show a change in practice. And I realized that that was what that was when I was a graduate student. So it's just a really cool opportunity to work with those growers and see that change in practice without knowing that's what it was back then. So this is sort of more on how they were able to come to their own decisions without us telling them they didn't need to spray the entire field. So I was really interested in using like GIS technologies and remote sensing to improve scouting and aid in decision making. So we had an undergrad at our extension center in Corpus Christi. He was in the geospatial sciences department and really interested in just using GIS platforms. So he was our GIS guy in the lab. We would digitize our map, which is a sort of very boring gray colored map here of our fields. You import that into our GPS device. You can import those random points we did. You can import that into this device as well. And then you can walk to the field to that exact spot and take samples in this device. And so they don't make these anymore, and I don't know why. But these were really easy to use because they relied on satellite data. So you didn't need phone service. So now we have ArcGIS Pro, which you can use on your phone, but it's not the same when you're in the middle of nowhere in a field and no service, unless you pre-download the map, but then that's file size and space. So you would take this data, you can enter the data directly into the device, and he created a Python script, so you get back to the lab, plug in the data, extract it, and then just run a script to create a map, which is just rudimentary here on the right-hand side, and we sort of color-coded it by the thresholds for a particular pest, whether it was in the cotton species or whether it was worms in sorghum, which is a little quick example on the next slide. And then we would send this to the growers along with an Excel sheet showing all of our data, how many plants we sampled, with the number of nymphs, number of adults, other injury ratings. So if we're looking at plant bugs, it was square retention. Stink bugs, we're doing green bull ratings as well. So they could sort of see the data for themselves, the numbers, but also in a sort of visual sense. And they could use it on their phone. So we send them as PDFs, and they can open up and just zoom in on their phone and look at it. And then here's just sort of an example of the broader scale sampling we were doing. So we would go out, check pretty much every other day, just do spot checking for various pests to see when they're approaching thresholds, so we can actually do that more intensive sampling to get my data. And this is just examples of those maps that we would create for different species and in sorghum. So that sort of quickly went through the pest side of things, but now as we move into the pollinators in the cotton agro ecosystems, or pollinator independent crops in general, so like what's out there, are they doing anything, what is a pollinator independent crop, and do they receive benefits from crops? So while I was doing all that sampling, and you can see with those maps, there's a lot of sampling, we're in the field a lot, Try to get there as early as possible because South Texas does get hot. So we were out there walking the fields, and we were going 300, 400 meters into that field, and I started to notice there were populations of bees in the system. And cotton is self-pollinating, doesn't need pollinators for fruit production. So I was interested, well, why are the bees here? And they weren't honeybees; they were native bees. And just started going down sort of this rabbit hole of looking into, well, what do the bees look like in the cotton system? Turns out there was not really much work done in the last 20 or 30 years. It was all done in the 60s, 70s. So I sort of convinced my advisor to let me take a little side project and turn it into a main sort of chapter of my dissertation. He wasn't very happy at first. I think he just did that to piss me off, and he just kept going. He's like, oh, no one cares about the bumblebees. He would call every bee a bumblebee, and he still does that today every time I see him. He's like, oh, how are the bumblebees? And I just shake my head. But he's like, okay. He's like, how about you put together a proposal, submit it to Sarah or Southern IPM for like their grad student program or their grad student grants. We submitted it twice, didn't get funded. The third time, it did get funded. And he's like, okay, yeah, you can do the project now. But I was already doing the project anyway, so I don't know why he really did that. But it was a good learning experience. So we were just talking about grants earlier. To submit that as a graduate student and get it funded for yourself, but it's really not your money. It's advisor's money, but not even his really either. So before we get into bees and cotton, let's just talk about pollination in crops. So we all know that pollination by insects is a critical ecosystem service necessary to produce most of our fruit crops, including those providing essential nutrients and fiber. I always like to include fiber in our sort of food security issues because we're all wearing cotton, I hope, at some point. And cotton comes from a plant. So I don't think a lot of people always think about cotton, where it comes from, and that potentially bees are in that system as well. But in terms of pollination dependence, not all crops are the same. So some crops rely on pollinators for fruit and seed production, which we call pollinator-dependent crops, and then others are self-compatible when pollinated or parthenocarpic, which we call pollinator-independent crops. So here's just some examples of pollinator-dependent crops. They're not perfect flowers. Some do have male-female parts, but they are self-incompatible. They have male-female plants or separate male-female flowers on the same plant. So pollination is required to produce fruits in those crops. And some examples are just almonds, apples, and cucurbits. And here's just images of their floral structure. So we have almonds and apples on the left here. So they do have both female-male parts, but they're, again, self-incompatible. And they need that pollination from a different variety even to fertilize and produce fruit. Whereas in cucurbits, that's our example of where they have separate male and female flowers on the same plant. And there's sort of an example of those structures. Now, on the pollinator independent cropping side of things, which what I'm interested in, are the pollinators with perfect flowers, which means they have both male-female structures on the same flower, and they are self-compatible. So they have the ability to self-pollinate, and pollination is not necessarily required to produce fruit in those crops. So some examples of those are cotton, soybean, canola, peanut, strawberries even. And then ones that really don't get any attention are wind-pollinated crops or grass-type crops, so like corn, rice, sorghum, and other grains. There's an example of the flower structures of sort of the main crops I work with there at the bottom. Now in terms of what pollinator-independent cropping agroecosystems look like, well, they can be diverse depending on where we are. So these are some examples of just areas I've been throughout grad school, postdocs, and my current location down in the southeast. So they're generally composed of a few row crops, sometimes monocultures that are variable in field sizes and geometries. We're going to go sort of through them here. And they can be diverse just depending on how we define diversity. For example, if we look at Texas in the bottom left here, if you look at the brick of red, the image of the brick of red, that's up in the Texas Panhandle, and all you see is red, which is cotton. Now, even within Texas, as we go down south, Nueces County, where I was down in South Texas, you can see a little more diversity in that system. We see a lot more fragmentation. We see different crop types, different field sizes. So we add corn and sorghum to that system. So I would say that is a little more diverse than, say, the Texas Panhandle. And then from there, I went to the Mid-South, or Mississippi Delta, where I did a brief postdoc. I also have a good friend and collaborator, or a couple good friends and collaborators out in the Mississippi Delta who are also working in pollinators in this system. And you can see even there we get more diverse, not only in terms of landscape structure, but we're adding soybean, we're adding a lot of rice to the system, inclusive of cotton and corn as well. And now if we go to the southeast, where I'm currently at, Florida Panhandle, South Georgia, South Alabama, it all relatively looks like that, where we have a lot smaller field structures, shapes, and we also have a lot of center pivot irrigation, so that also we see a lot of circular fields compared to sort of our rectangular, squarish shapes in other parts of the country. So this provides sort of an interesting way to look at things in the Florida Panhandle and how those sort of that landscape structure affects pest and pollinator populations. And I think this is really a really diverse area, at least in terms of agronomic crops or row crops, because Florida, at least we have a lot of semi-natural habitat and a lot of swampy areas, just a lot of different vegetation that surrounds a lot of our agricultural fields. Now, if you look at something like the Midwest, no offense to the people from the Midwest or doing work in the Midwest, it's relatively boring when you look at it. It's all soybean and corn. So, and those are important that we have some good people doing bee work, like Matt O'Neill at Iowa State, doing really good bee work in soybeans, but it's just, I don't like corn. That's my, my thing. Now, finally, we're getting towards more, now what bees in these systems? Are they managed, unmanaged bees? You can call them apis, non-apis, wild honeybees. I think managed and unmanaged is a good term to use. I've used all of them. It's just sort of what goes with the context. So in terms of our managed, they're mainly social species. We're all familiar with honeybees, Apis mellifera. They're technically an invasive species, but don't say that at a group of beekeepers. To a group of beekeepers, they're eusocial, have a broad diet breadth, but they're not always the most efficient pollinator. And then we have a lot of other managed wild bees, for example, is why it's sort of hard to use wild and non-wild bees. Things like Bombus terrestris, they're really good. You can buy those now and get them shipped to you. They're really good in greenhouses. There's also multiple species of Bombus across the world that are sort of managed in that type of way. And then we have our sort of other specialty managed bees that are also wild. Those being the alfalfa leafcutter bee, so Megachile rotundata. I think y'all should see quite a lot of those, especially in the blueberries and fruit and tree orchards. And then the alkali bee, which is a really special case, and I'll talk about that in a second. And then mason bees, so it's multiple osmium species, so like blue orchard bee and things with names like that are generally mason bees that are also managed. And here's just different examples of what management of those species, whether they're wild or honeybees. So honeybees are obviously the easiest to, say, transport around the country because you can take them to crop, from Florida to California, depending on what needs pollination services. And then we get into our orchard or leafcutter bees, where they're also a little more easier to manage as well and transport. So they're cavity nesters, so they need somewhere to nest. So what people have done, they create little bee boxes, which is basically a block of wood, pre-drilled holes in it, relative to their body size. So they actually use that hole, or people use like bamboo sticks that are hollowed out. And what they would do is set these out. They get a population that would go in, make their nest, and then once the adults leave, they would put those in the incubator, put them in the fridge, and they'll sort of overwinter. And then they take those boxes out, put them out the next season to provide pollination services to whatever crop needed it. And then so for the alkali bee, which was sort of a special, like unique case of management, I think it's our only ground nesting bee that's sort of managed. And what's actually cool is that growers out in the Pacific Northwest for alfalfa seed production, they will actually set aside portions of their land that are prime nesting sites for this particular bee. And they don't put that into production to sort of entice these bees to go and colonize that nest or colonize that ground. They sort of nest in aggregations. You can kind of see that with the holes there. But it's relatively hard to do that because you got to convince the growers to take away that land from production, which in terms of yield is money, so they kind of don't like that. But in the service they provide for that particular system, it works quite well because it is needed to produce alfalfa seed. Then we get to our unmanaged species, which was I'm really interested in, or wild native species, which are very diverse in natural history, resource requirements. They can be generalists, specialists, mostly solitary, some communal aggregations, some semi-social on the verge of sociality in these species, and they're mostly ground nesting. We got cavity nesters, so our leaf cutters, our megachilids are sort of cavity nesters, but a lot of the species I'm going to be talking about or we see in row crops are our ground nesting bees, and sometimes they are more effective pollinators. And then here's just an example of how diverse these wild bees can be. You can have very, very tiny, the world's smallest bee, over there on the left-hand side. It's a Perdita species, to our very large bees, or our carpenter bees, Xylocopa species, on the far right-hand side on the bottom there. So now what about the relationships of bees and pollinator independent crops? Are there benefits to bees from the crops, or are there benefits from the bees to the crops? And so one thing that all these crops I've mentioned have in common, in addition to being self-pollinating, is that they are all what we call mass flowering plants. So think cotton, soybean, canola, peanut, corn. Technically as a flower produces an abundance of pollen. For example, soybean produces 800 florets per plant, up to 500,000 per acre. So that's potentially a tremendous resource for bees in a system. For example, since I work in cotton, I like to point out that the cotton, each one of these fluffy white bolls was at one point a time a flower over the season. So not only is it all at a lot of resources all at once, it's for a consistent duration. So cotton flowers for about 6 to 8 weeks. So from late June to middle of August, you have this source of floral nectar and pollen that the bees can potentially use in the system. But we don't know is, say, how nutritious that pollen is, and are the bees actually going to it. So some people have done studies recently. So this is a table from a paper by Silman et al., who comes out of Maggie Couvillon's group out of Virginia Tech. We were just talking about waggle dances earlier in Cesar's lab meeting. They actually analyzed a bunch of waggle dances from honeybees in a pollinator independent cropping system. But I think they used sort of an AI method in which they just video recorded and let the AI sort of analyze where they were going or get a sense of where they were going. They would map it out, and they go ground truth that data to see what was actually there. And it turns out that honeybees frequently visited row crops during bloom, when the crops were in bloom, and peanut was the more favorable, which was sort of interesting to me because I haven't relatively thought about peanut or bees and peanut, and hadn't seen any literature in bees and peanut. And there has been a couple since the 2020 where people are actually starting to look into bees in these systems. So it seems that row crops can provide a forage source for honeybees, but like I mentioned earlier, what are they foraging for exactly? Is it nectar? Is it pollen? And then what is the nutritional value of that nectar and pollen? So now we can get into the cotton system, or sort of the data that I've been involved in. So what's going on in the cotton system? Are we talking about honeybees, or are we talking about wild bees? So honeybees in cotton, honeybees generally don't like cotton. The pollen is too big, and it sort of interferes with their ability to pack it on their body to take back to the hive. I had a video, I couldn't find it to put in here, but I would see honeybees in cotton this year, and they would immediately go to a leaf and then brush all the pollen off of themselves. So they really don't like pollen. But then talking with people, some beekeepers in North Carolina, they're actually keeping their honeybees in cotton fields to produce cotton honey. I don't think that would taste good, but that's what they do. And then I met some beekeepers from Israel saying that they use beekeepers for hybrid cottonseed production. And they said their bees love the pollen. So it's sort of just interesting. However, in Texas, we didn't see any honeybees in our system. In Australia, they also see bees in their system, which is why I have this review study here, that they looked at bees in Australian cotton, and they found that 31 species looked at bees in cotton, and some indicated a positive benefit from honeybees. So I was really interested in, okay, well, what's going on in my system, and are there actually benefits to bees? And I'll talk about Florida in a second, because again, it's a little different now, too, that I got data this year, in which we did see a lot of honeybees in the Florida Panhandle. Now, on the wild bee side of things, there's even less information on, say, the pollination benefits from wild bees. They're positive, no effects. Most of the data we found did indicate some positive effect or just no effect. Diversity-wise, some cases we know the diversity of bees in this system, and a lot we don't know. So again, there's pretty much no sort of diversity data across the entire Southeast, especially in our row crop systems. But one genus that does come up in a lot of the literature is Melisodes, so our longhorn bees. Pretty much come up in every study that looks at bees in these row cropping systems. So there's multiple papers in Texas, Mississippi Delta, Georgia, Brazil. So it's not only a U.S. thing. There's also a Old World sister genus found visiting cotton in Africa as well. So it seems to be sort of worldwide that this particular genus doesn't seem to mind cotton flowers. But they are generalists, so that may be some part of the explanation. But you do see those most abundant in cotton. So what was going on in the system where I was doing my work down in South Texas? So as we were taking that pest data, I was thinking, well, how do sort of landscape factors affect bee diversity? And with bees, you can do a little more sort of landscape level approaches, but I'm not going to be showing too much of that just for the sake of time. So we sort of came up with the same concept, does landscape structure, what's next to a field, affect the bee diversity within that field. So we did sort of same sampling structure, but we used bee bowls. So we placed 2 pairs of bee bowls at each side of the field, and we were looking at different crop interfaces. So with bees, they do have the ability to travel a little further to forage, so we needed to make sure our sampling sites were spread apart, and then we also put traps out in the semi-natural habitat alone to see if there's any differences from sort of natural area or the cropping habitat. And then we did sort of a control trap as well, in which we found a field that was at least 200 to 300 meters away from semi-natural habitat, just to see if the bees were going that far, to sort of hypothesize, if they're out here in the middle of nowhere with no nesting sources or alternative sources for food, then they must be sort of nesting in the field. That is what we still don't know. So what did we see from all this sampling? So we did 2 years of sampling. We significantly increased our sampling effort the second year, which is why there's this difference from 2017 to 2018. But in total, we ended up with about 5,500 individual specimens. So excluding Apis mellifera, that accounted for 33 species across 3 families and 13 genera. And then we had 2 dominant genera, so one being Melisodes, which I sort of mentioned or sort of preluded to, and 2 species of that was Melisodes tepanica, which tepanica is by far the most common, and then Melisodes communis was also found. And then our second most abundant genera, or is more of a species group, was Dialictus, so that's a subgenus of Lasioglossum. Those are our little small sweat bees. They're extremely difficult to identify, so we did identify some, but then just identify the rest of morphospecies, and I just consider them as a Dialictus complex. Now in terms of landscape level effects, so this is just species richness in this figure up here on the top right. You can see we're kind of even across our species richness, across our interface types. So we're seeing pretty much all the species at some point in time in the field, but it's the abundance that's what's going to differ, in which I'm going to show you in the next slide. So just the field interface influences abundance of our most dominant species, and I'm just going to focus on the most dominant species because that's where we can really see sort of effects of landscape structure on them. So if we look at Melisodes there on the left-hand side, they seem to have more of affinity to our cropping interfaces. So this yellow bar is the interface of cotton and sorghum field, and you can see that we found significantly more bees at that interface compared to, say, the natural habitat alone, or the interface of cotton that was next to semi-natural habitat. And then our other interfaces, whether it was cotton next to cotton, also 200 meters away from semi-natural habitat, was sort of marginal differences between the two. So they appeared to sort of like those crop-crop interfaces compared to, say, our Dialictus, which gets a little more interesting, and then it's sort of opposite. So we saw more Dialictus abundance at the interface of cotton and semi-natural habitat. But what's even more interesting is that there was more at that interface than, say, the natural habitat alone. So if you think about in terms of what's going on in the summer, and in terms of bee biology, so generally foraging distance is relative to body size. So Melisodes is a relatively larger bee, it's a little bit bigger than a honeybee. They can go maybe about 500 meters foraging range, compared to, say, something tiny like Dialictus. It's going to have a significantly less foraging range. So my sort of hypothesis is, and I've actually seen some current folks working on it from Auburn, is that Dialictus seems to like cotton, but it's only found in the cotton that's next to sort of either there's weedy habitats or semi-natural habitat where they can nest in, or there's alternative forage for them before cotton's planted. So that sort of just got me thinking, like, our Melisodes, the Melisodes have to be nesting in the field, especially when they're, say, 200 to 500 meters away from any potential semi-natural habitat where they would nest, whereas the Dialictus are probably nesting in that semi-natural habitat and not the cotton itself. But again, we just don't know because there's not much work done on it. Now, at the time, I had a friend and collaborator in Mississippi Delta also looking at bees in her cotton system. So we decided to share our diversity data and sort of compare and contrast the diversity across the regions. And it turns out there wasn't much differences. They look pretty similar. Obviously, Melisodes is this giant red portion of the pie chart here. There's the dominant genus in their system as well. They had a little more diversity in terms of, say, species. They had all 4 bee families and a little more genera diversity compared to Texas, in which it was all basically Melisodes or that Dialictus complex. So there's relatively similar in species composition for somewhere that was, say, a few thousand miles apart. So seems to be similar bee diversity in these systems. So I'm sort of interested in what we see in Florida, and we'll talk about that here soon. So we see that there seems to be some sort of diversity and abundance of bees in the system, or in cotton systems. So I was interested, well, does cotton benefit from pollination by these bees or from bees? So like we mentioned before, we know cotton has perfect flowers. They can self-pollinate. Flowers are only open for a short period of time. You know, the pollen's too large to even be carried from plant to plant by wind. So if there's any cross-pollination going to occur, it's going to need the assistance of pollinators. So I was looking into literature, this is back in 2016, and there wasn't really much work done. There were some correlative studies where they would go take a population of bees and sort of take yield samples of cotton in that area and to see if there was any relationship between yield and the presence of bees, or there was a lot of hand crossing data. So this group out of UT Austin did a hand crossing experiment in which they saw cotton benefited from a 17% increase in seed cotton weights when it was hand crossed. Now I saw that and I was like, well, what happens if we actually force bees onto the plant, or sort of do a forced pollination experiment? So we did a little pollinator exclusion/inclusion experiment in which we caged Melisodes tepanica onto cotton plants to see if there was actually any differences potentially from bee visitation on yield. So we had 4 treatments: one caged cotton plants that were excluded from pollinators, caged cotton plants which we added pollinators, and then caged hand-crossed treatment, and then of course uncaged plants exposed to whatever was in the system at the time. Now for our bee treatment, so Melisodes tepanica was so abundant, we would go out to the field, or that field we were working on, and just hand catch bees, bring them back to the lab, let them chill to the late afternoon, and then put them in the cages late afternoon so they don't get any heat stress. And we leave them there 48 hours. After 48 hours, we do it again. We did that for a period of 2 weeks, and then sort of analyzed our data. And then for our hand crossing treatments, so what a lot of people didn't do in the past is that with cotton being self-pollinating, and sort of anthesis occurs really quickly, that you have to emasculate the plant before you hand cross, so they make you ensure that you are actually getting a true cross, and it's not self-pollinating before you make that outcross from pollen from an adjacent plant. So we would tag individual bowls of blooms in the candle stage. Cotton people have weird lingo for things, so this is what we call candles. Kind of looks like a candle. I don't know, maybe been years of just being called that, sort of got that in my head. But those are called candles. So what we would do the day before we were going to expose our plants to these treatments, we would emasculate the flowers, so we remove the petals, remove the anthers, and then tag all those bowls. And then our non-hand cross treatments, we would tag bowls at that stage as well, because cotton is indeterminate growth. So we want to make sure we're comparing the same age and size of bowls on the plant. Now, what happened after all of these, across those 2 years with that experiment, so this is just seed cotton weights, and that is the inclusion of lint and seed before ginning. I'm just going to talk about this for sake of time. I know you don't see the same figure at different sizes for 5 slides in a row. But what we did see is that bowls produced from flowers that were exposed to Melisodes tepanica, so the orange bar, and flowers produced from bowls that were hand-crossed, and bowls that were produced from flowers that were just uncaged, all had significantly higher seed cotton weights than our excluded treatment. So some form of pollination did increase yield, meaning that cotton potentially does benefit from pollination, whether it's by insects or by hand. Now, what does that mean in terms of yield? So, and this percentage is just in reference to the bee treatment. So on average, we saw a 32% increase in seed cotton weight, but only on those bowls exposed to Melisodes tepanica. Now, when you think about cotton, that's just on one bowl that we expose to a bee. So you have to take the whole plant average, and there could be 100 bowls on a plant. So across the whole plant, that turns out to be about a 12.8 or 13% increase across the whole plant, which is, to me, significant given the fact that cotton self-pollinating and these bees are already in the system. Now, I talked about this to that same management team, and they're like, well, how can we get more bees? And that I don't know the answer for. We still don't know the answer for, but they saw that data and they're just interested in, well, hey, I can get 13% more yield if I have bees in the system. So they're interested in that side of things. That's sort of my philosophy now, is sort of grower education on pollinators in their systems, and how they can potentially benefit from it. Because a lot of people think of bees as pests, believe it or not. in terms when you mentioning bees in ag, they're worried about insecticide usage and what are they going to ban next. But I'm trying to change their view on that. Now let's talk a little bit less about cotton, just very briefly. Soybean, we kind of see the same sort of relationships, very diverse population of bees in the soybean community. So we put together a review paper in which we tried to put all the literature on pollinator independent crops, studies that looked at wild bees and benefits to those crops in one place. And we found that there was a maximum of 68 species found in one study in a soybean system. But what's consistent is that Melisodes and Dialictus were common as well, pretty much in every study that looked at these bees in soybean systems. So very similar to cotton. And there's been more work done in soybean for some reason in terms of nectar and pollen sources. So even in these studies, say 28 and 39% of bees over 2 years, this is from Matt O'Neill's group, were carrying soybean pollen, and that was consistent with older literature as well. So the bees are visiting cotton or visiting soybean for floral resources. But soybeans are highly variable across varieties in terms of nectar production, how much they produce, or some don't produce any at all. So there's, again, still a need of work to be done there. And then for cotton, we just don't know. Now, are there benefits to soybean yield? So we found about 20 studies that did look at this. And when they did find a increase in yield, it ranged anywhere from a 6 to 20% increase when soybeans were exposed to bees. So there's definitely some similarities across these crops. There's just a lot of unknowns. Now, what about benefits to bees? What can these crops provide to bees in the system? So if we think about cotton, I mentioned again that each one of these bowls was at one point in time a flower. Cotton has a lot of extra floral nectaries, which is an example here on the bottom right. You can see a visible drop of nectar, and that's on every flower, on every plant. So that's going to be potentially a lot of nectar availability across a period of the summer in which nothing else is flowering. And they also produce an abundance of pollen. I forgot this little video of a sweat bee sort of foraging at the bottom of that flower, likely getting nectar. So the floral nectar sort of evaporates in the heat and crystallizes, so they get down in there and collect those crystals, rehydrate them, and then use that for their fuel resource. Now on peanut, they produce pollen only. Honeybees seem to like it. There isn't relatively no work done on the nutritional value of that, or just bees collecting peanut pollen, aside from those that one study I did talk about. But that was honeybees, so we don't know on the wild bee side of things on what is visiting peanut and are they using that pollen. But it turns out they are, because you can see the honeybee there on the peanut flower. I don't know if you all ever seen a peanut flower, but that's what a peanut flower looks like. And that those sort of the main male-female parts there are sort of enclosed in a little, I don't know how to describe it, like a little clamshell. And when a pollinator visits it, it opens. So I walked a lot of peanut fields this year, and I saw a lot of that, meaning that the bees were there. And you can see a honeybee lower in that canopy visiting another flower after it just finished getting pollen from that flower. So they are using that bee. So what's going on, say, in bees in Florida crops? So this past season, we did a lot of sampling, did a lot of bee bowls, and we also did some visual observations. So we walked 50-meter transects across peanut and cotton, and we observed 516 visitations. So we were looking at least in cotton, we were looking at what are they doing in that flower? Are they foraging for nectar, pollen, both? Are they nectar robbing? Are they only going to those extra floral nectaries? And then in peanut, we were just seeing if they actually visited the flower and they spent time there. So what we saw just from observations and visual IDs is that there was a lot of honeybees across both cotton and peanut. But interestingly, I don't have the data in a graph to show you, but pretty much, I'd say 95% of the honeybees visiting cotton flowers were nectar robbing, or visiting that extra floral nectary and not the flower itself. And if they were in the flower, they likely just flew in there by accident. And so I'm still working with this data set. I think it's going to be really cool, specifically with cotton, because as we sampled throughout the years, what position are they visiting, what node on the plant, what branch on the plant were they visiting, and if that makes any difference as far as visitation and yield benefits. Now, what else are they doing in cotton? I mentioned that we were unsure if they are nesting in the cotton or not. I hope this doesn't. Okay, we're good. So I was never able to find nests when I was in South Texas, or Mississippi for that matter. But turns this year, I didn't actually find this. the rest of the faculty and crew knew I was interested in bees, so they were out. This was a aggregation of Sphaspora species. We thought it was Melisodes just from briefly looking at it, but it ended up being Sphaspora after we caught a few and tried to ID them. They actually found this. He gave me a call. He's like, hey, I got a swarm of something out here. You should probably come take a look at it. I'm worried they're hornets. And I was like, well, are you standing next to them? And he's like, yeah. And I'm like, well, they're probably not hornets, so you're safe for that. And so I went out there, and it was really cool because I have never seen anything like this. Like, there's literally hundreds of male bees flying over these holes. There's about 15 to 20 nests within like one plot of a treatment and a nitrogen rate study. And like you could just walk through that. The wild bees are pretty docile. You can probably pick them up, especially if they're males. But they're all waiting for a female to come out and sort of fight for each other to mate. And there's sort of a little image of that there. And these nests are pretty, and so we don't know where they're nesting, and there's even less known about nesting structure. So I tried my best to sort of dig down, dig up a nest, sort of drew it to get a little diagram of, well, what does a Sphaspora nest look like, because there's nothing done on it. They actually go down pretty deep, so we dug up a few. So initially it goes straight down for about 3 inches, and then it sort of slightly curves, and it went down to about 16 to 29 inches, which is quite far, I think, especially in the soil types. Like this orangey dirt is clay, so they're going through clay as well, which I think is kind of crazy given it's a little, well, not a little bee, but it's a bee digging almost 2 feet down into the soil. And there's also seems to be a little ecosystem of bees. So this is a parasite, this Tripiolis species there. It's a parasite of Sphaspora and Melisodes bees. So there's a lot of interactions going on within a highly managed research farm trial. So sort of a cool experience seeing that, because a lot of people in literature say they don't nest in the agricultural field, and that it leads to implications like, well, is tillage good for bees? Is it bad for bees? Everyone thinks tillage is bad for bees, but that was strip tilled. In Texas, it was all conventional till, deep tillage, so it seems to be not affecting bees, maybe, I don't know. And that's the line I've talked to a few of you about. It's hard to balance that relationship of bees in agricultural systems. So why does this all matter? Well, when you look at these systems and you look at the amount of acreage of pollinator independent crops in the system, you know, this is a lot of potential floral resources to support wild bees throughout the season, especially when there's nothing else growing. So it's sort of interested in seeing, well, how do we bolster populations of these bees in the systems, and what's the importance of having sort of crop diversity in a landscape to provide forage for bees? And that's not even talking about the pest side of things as well, because that's also going to have implications on the pest side of things, which is why I sort of have this broad interest in studying all, whether it's pollinators, natural enemies, and pests, to see how it all works in a system. And as far as like what's next, like I mentioned, we still don't know a lot about the natural history of these bees, of the dominant visitors, for example, and then the nutritional content of that floral nectar and pollen, and its implications for bee health. And I think Pierre Lau is coming next month, who is my collaborator handling the nutrition side of the stuff on this project, and he is probably going to talk about more on the nectar and pollen nutrition in these crops. So sort of a little setup that kind of worked out well for me to come first and then him talk about the more detailed stuff. So with that, I'd like to thank Chloe for the invitation and for y'all hosting me, and we'll take questions. Don't comment. I put my cell phone there for growers to reach out.
So that clock's a little wrong, but
Yeah, I don't know
we
how-
have time for a couple
Okay.
questions.
I wasn't sure on the first part. It's actually on time-ish.
It's like column thing, it always holds.
Yeah, it's I'm on
It's an
time.
extension thing.
Yeah, I'm on time though.
No, you did this in graph.
He was destined to be.
Yeah. Yeah, hell yes.
Are cotton, is cotton clonally propagated?
You know, I don't know how too much on the seed production side. I should.
Like are the different cultivars?
Are you in
like,
terms
they,
of
they,
like?
They don't require the pollination, like I'm sure.
No.
Self-compatible?
Well, they
Yeah,
are
they're self-compatible, but you're talking about like where you get the seed. Are they all the same?
From like cutting or something?
No, it's all seed. So it's all you're producing cotton seed from cotton plants.
And
That's done like a lot in Arizona.
So the fact that there might be pollination means that you don't have pure.
Yeah, well, yeah, and I talked to breeders about that. So you know, they are the same cultivar, but there's still genetic differences in those plants next to it, even if it's the same cultivar. But I don't know like the specific details on that, and I need to talk to the breeder again. But she was really interested, and so they actually bred out a lot of nectaries too in cotton breeding. But things like Pima cotton, that's the really fancy cotton, the really soft cotton, that produces a lot of floral nectar or extra floral nectar. And that she was saying that you could walk into that field and you can smell it. So there's a lot of interesting things on terms of the genetics side of
Do they
the
grow
plant.
each like block of? Do they grow like the same cultivar next to each other? So like when you did that cotton interface, were they the same?
Yeah, so
cultivar?
usually they're the same cultivar, and I was grad school, so we didn't get that information. So now I always take what cultivar you're planting or what you're actually sampling when it's an on-farm study. But generally, like one field will be the same cultivar. Some peanuts I see a little more where they'll split cultivars, but cotton-wise, it's usually the same.
Did you, I know this was back in grad school, but when you were doing the landscape analysis and kind of looking at the edge effects that you saw
Mhm.
with is that plant bugs?
Yeah, plant
Yeah.
bugs.
Did the size of the field, I mean, we talked about this
Yeah,
yesterday.
yeah.
The size of the field between your studies and my studies are really different. But when you did your analysis, were you looking at the size of the field to see how that affected the edge?
No, like in terms of like how much edge there was. No, I didn't do that, and one because we didn't sample on fields that were small to like compare differences.
Okay.
And those fields, there actually is some variations. Like it's a good idea that should potentially go back to do, but they were all about 700 to 1,200 acres, so they're relatively similar. So I just didn't think there was going to be differences, but there could.
We've done this at 5, 10, 20
Yeah.
acres, which is big for us. That's a lot of peaches.
Yeah.
And we do see the edge effect doesn't disappear, but the management starts to break down after a certain point. If you only do a border spray,
Yeah.
it starts to break down just because it's.
Interesting.
Well, the percent edge
Yeah,
changes.
goes down.
So,
Or up.
in a 5-acre block, it's about 25%, but in a 20-acre block, it's a smaller percentage that you're managing if you're doing a border spray. So overall, we don't see, the management isn't as effective as when you get to a bigger plot size.
Yeah.
You have to do a few more rescue treatments.
I had a question about that, and forgive me, I missed the beginning, but the fact that you saw more of those green bugs, I don't remember the names of the actual pests.
The Verdes, or yeah.
Does that mean that they're not really feeding on the crop?
Well, they are feeding on the crop, but I think a lot of those plant bug species in cotton, they don't like cotton. That's just what's there. So they do like. I mean, they prefer their wild hosts, but like in the season, their wild hosts and nest like late spring. So then cotton's there. So it's mainly I think with that particular species and that Verde plant bug even more so. Their primary host was like it's a succulent type plant. So that's what you see a lot along of our like rivers and creeks in South Texas. That plant grew there. So that's likely why we're seeing that higher. Well, when you're covering the whole county level in South Texas, yeah. I mean, that's one solution, but it's hard to get to those areas at least.
Well, the natural habitats also host a lot of.
Yeah, then you'll get.
But also pests.
Yeah.
Also pests. The natural habitats have a lot of diversity.
The bee people will come after you if you want to get rid of that. Let's go with someone else and see how else too many questions.
Quick clarification question, because you said that the flowers of the cotton plant are open for about 8 to 11 hours, and then you said they were short. Is that like 8 to 11 hours total, or like per day?
So like they're viable, I would say, for 8 to 11 hours, but still like I see bowls produced from flowers that don't open, so that anthesis, that pollination occurs quickly. But they stay open, so they'll change too. I had the picture where they went from white to red, so they're there for a couple days and they'll fall off. But as far as when pollination takes place, it is within that short period of time. Yes.
There's a good amount of native bees that use old nests of other
Mhm.
bees. Do you think the tillage might impact seeing
So that's a tricky question, and so it definitely will affect some bees. So it's when I say it's like in terms of the bees I was looking at, those Melisode species, the generalists, seem to be kind of persistent in those systems. But some of the smaller bees, some of the more rare bees that we didn't see, it's going to really affect their nesting habitats.
Yeah, I was just wondering because it's like, I wonder if there would be more of these like second year bees that use.
Yeah, so that's why I'm curious with the nesting we found. Like they should be back just based off of the way bees work. Like they should go back and nest in that plot again. So I'll see this year if they come back, and if it's tilled, I don't know what they're going to do with that plot. But that's what I'm interested in too, as far as the soil.
That's all the time we have for questions.
Okay.
But Isaac will be around.
All right, thanks again. Yeah, I'll be around the rest of the day if anyone's interested in talking about anything. Oh shit, were there questions online?
Huh?
Oh. I was like, were there questions online?
I never look at that.
We are going next door to our regular.
Did you see Grayson was like doing an impression of the bee in the video?
It's cute.
What? Oh, I
The
was
one
gonna.
coming out of the hole.
I have a bee doing dancing or shaking its butt to like some Meg the Stallion. I was like, that's probably
Do you
not. That's
need,
probably not
I
appropriate.
don't think
Yeah.
do you need the bathroom or have
Don't
like
forget
around?
your
No, no, just
pen
me.
drive, your thumb drive.
Do you want me to go and get you coffee?
There's coffee in the back.
I'll get some.
Well, like Starbucks or something.
No, I'll be fine.
We have soda. Yeah.
He doesn't drink soda.
No, I'll be fine.
He only drinks
I mean, if you have
carbonated
coffee
drinks with his
available, I'll
beer.
take some. But
I
Don't
pretty
go
much
eat
go
it.
from coffee to water to wine, so
yeah,
you know, I
exactly.
get it.
Comfort
Yeah.
water.
He goes from
Water,
beer to
beer,
coffee to
liquor.
beer to beer to cocktail.
Yeah, just don't forget the.
Yeah, that's Chloe's.
Did
Chloe's
you-
take care of
Yeah, so he doesn't care if
Brandon's.
he loses it.
He forgets it.